Hello, this is Dr. Komal Acharya welcoming you on Biobasics. This time I am going to talk about gene regulation and the Operon system in prokaryotes, which is in continuation with the previous video lecture, Gene Regulation Part 1. Okay, so let's begin. The prokaryotic cell contains thousands of genes which are expressed through the production of proteins or enzymes, and from these enzymes, some enzymes are needed by a cell all the times, like enzymes needed in central metabolic pathway. Okay, so such enzymes are known as constitutive enzymes, and genes encoding such enzymes or proteins are known as housekeeping genes. Expression of these genes occur constitutively, which means they are expressed constantly and not needed to be regulated. Right, whereas there are some other enzymes which are needed only at certain time under certain conditions. These enzymes are known as inducible enzymes. And because the genes encoding such enzymes are expressed only when needed, or we can say only when they are induced, it is known as inducible gene. Constant expression of such genes is wasteful, so their expression is needed to be regulated. Now, as we have discussed in last video lecture, cell carry out the gene regulation at different levels. Okay, so this time we are going to talk about the transcription level regulation, particularly induction and repression mechanism. Now the most common step or process at which the gene expression is regulated is transcription initiation. Regulation can also occur at elongation and termination steps of the transcription, but regulation at transcription initiation is energetically more active, more efficient. But before going into detail about different regulation mechanisms, let's talk about some important terminology. First of all, induction. Induction is a process that induces or promotes the transcription of gene. Repression. It is a process that represses or inhibits the expression of gene and so the enzyme or protein synthesis. Repressor. It is a gene regulating protein that binds to the target sequence of the DNA and prevents the transcription process. Operator. It is a specific site on the DNA to which the repressor protein binds, and it is always located downstream of the promoter and closer to the coding region. Now let's talk about the inducible and repressible mechanism. The most studied induction and repression mechanism is the Operon model, which was proposed by French scientists Jacob and Monod in 1961. They established the Operon theory, which explains the regulation of gene expression at transcription level. Basically, Operon is a sequence of closely associated genes that regulate enzyme or protein production. In general, Operon is composed of two things. One is structural site, another one is regulatory site. Structural site includes one or more structural genes which carry the information for protein synthesis, while the regulatory site includes promoter and operator which controls the transcription or expression of these structural genes. It also includes the regulatory gene that works in coordination with the operon, but it might be located at some distance. Now, this operon theory is applicable for both the eukaryotes and prokaryotes. But as far as the prokaryotes are concerned, several structural genes are regulated by one single operon. And one such classic and most studied example of lactose operon of this operon model is lactose operon. Lactose operon is a typical operon that controls the catabolic pathway in E. coli. Now, why this lactose operon is needed, or what's the exact function of it? So look at here. Lactose is one of the many organic molecules that can be used by the E. coli as carbon and energy source. But if lactose is not available, then what will happen? The enzymes needed for lactose metabolism are not needed, and so the lactose operon will be turned off. But if the lactose is available to the bacteria, then enzymes for lactose metabolism will be needed, and so the lactose operon will be turned on. Right. So the expression of genes encoding lactose metabolizing enzymes is wasteful if the lactose is not available. So the cell expresses these genes only when lactose is available. Now let's talk about the lactose operon structure. As all other operons, it has structural site and regulatory site. As far as structural site is concerned, there are three different genes, LAC-Z, LAC-Y and LAC-A. Each encodes for different enzymes. First of all, LAC-Z, which encodes for beta-galactosidase enzyme. Once the lactose is inside the cell, this beta-galactosidase performs two functions. One, it breaks down the lactose molecule into galactose and glucose. And second function is that it performs isomerization of lactose into allolactose. So this was the function of beta-galactosidase enzyme. Now LAC-Y encodes for lactose permease enzyme. This lactose permease ensures the transportation of lactose molecule through the semi-permeable membrane of the bacterial cell to inside the bacterial cell. Last one is LAC-A, which encodes for beta-galactoside transacetylase. Exact function of this enzyme is still not clear, but roughly we can say that this enzyme plays a role in detoxification of beta-galactoside by transferring the acetyl group from acetyl coenzyme A to beta-galactoside. Okay, so these are the structural genes. Now let's talk about the regulatory site. It contains the promoter and operator region. Let's talk about operator first. Operator region is located between promoter and the structural genes. Okay, basically LAC repressor binds to it and negatively controls the transcription process of operon. Now LAC operon has two types of operator sites. Primary operator site O1 and secondary operator sites O2 and O3. Okay. Now next one is promoter. This is the site on the DNA to which the RNA polymerase bind to initiate the transcription process. Apart from that, it also contains repressor gene, or we can say the regulator gene I, which encodes for the LAC repressor protein. Let's talk about LAC repressor now. LAC repressor protein is a tetramer. Okay, it is a product of regulatory gene I, which is a negative regulator as it inhibits the transcription process. If you look at the structure of the LAC repressor, it is a tetramer made up of 4 different subunits, 1, 2, 3 and 4. Each subunit has DNA binding domain. Right. It has 2 recognition sites. One is DNA binding site, which recognizes the operator sequence present on the DNA. This is the operator DNA sequence and this is the DNA binding site. And another one is the allosteric site that binds with the inducer like allolactose. Why it has allosteric site? Because the LAC repressor is an allosteric molecule, which means that the LAC repressor binds with the inducer and the conformation of the LAC repressor protein is changed. Okay, so an allosteric molecule is a molecule which shape can be changed according to the substrate to which it binds. So once the LAC repressor attached to the inducer molecule, then the conformational changes occur in the repressor protein, which makes it inactive, which makes it unable to bind with the operator region. Right. Now another important thing. There are 2 operator sites, as I told you, primary and secondary operator site. Now LAC repressor is a tetramer which has 2 dimers. So one dimer will attach with the primary operator site O1 and then the second dimer will attach with the any of these secondary operator sites O2 or O3. If the dimer binds with the O2, then it will create a band like this in DNA. If it binds with the O3 operator site, then it will create a loop like this in DNA. So in any case, it binds with the LAC repressor protein will bind with the DNA and it will form a loop like structure. Okay. Now, after going through all these important molecules or important components of the LAC operon, let's see how it works. But before going into detail about Lac-Operon system, for better understanding, let's take a quick review of transcription process. How the transcription begins. Transcription begins when the RNA polymerase bind to this promoter region. Then it will unwind the double helical structure of the DNA and it will continue the transcription of the double helical structure of the DNA. But basically, for initiating the transcription, RNA polymerase has to bind to this promoter region. Okay, now let's get back to the Operon system. How it works. Now see, when lactose is not available, then what will happen? The regulatory gene will be expressed and it will synthesize the Lac repressor protein. Okay, this Lac repressor protein will bind to the operator region. This is how it binds. Now, binding of this Lac repressor protein to the operator site is also two-step process. In the first step, the Lac repressor protein binds non-specifically to the DNA, and in the second step, it will slide along the DNA and till it reaches to the operator site. Now, it will bind to the operator sites like this way. In any case, it will create a loop-like structure in the DNA. So, there will be two possibilities. Due to the bending of the DNA at promoter site, the RNA polymerase cannot get access to this promoter region. In second possibility, RNA polymerase cannot move towards the coding region. So ultimately, in any case, the transcription of the structural genes will not occur. Right. This is the case of lactose unavailability. If the lactose is available, then what will happen? The Lac repressor protein has been synthesized. Now, lactose is available, so beta-galactosidase enzyme will convert this lactose into allolactose and it will act as inducer. It will bind with the Lac repressor protein and converts it in inactive form. So it cannot bind to the operator site. So the RNA polymerase will attach to the Lac promoter region and then it will carry on the transcription of these structural genes, and so the lactose metabolizing enzymes will be synthesized. So this is how the Lac-Operon works. But there is another phenomenon. If the bacteria, here particularly E. coli, are provided with different carbon sources, it will show preferences among potential carbon sources. If glucose is present with other sources like lactose, then E. coli will use glucose preferably. Why? Because it can be used instantly and doesn't require induction of Operon. So when glucose is present, lactose metabolism will be repressed. Synthesis of lactose metabolizing enzymes gets inhibited. This inhibition in presence of glucose is known as glucose effect, or more precisely we can say catabolic repression. Now Monod observed this phenomena when he provided both the glucose and lactose to the E. coli culture. He observed that bacteria utilizes glucose first and represses the lactose metabolism meanwhile. And once the glucose has been utilized, it will start using lactose, which results in such type of dioxic growth. So when the glucose is present, a specific regulatory system ensures that Lac-Operon remains inactive because lactose metabolizing enzymes are not needed at that time because glucose is there. And this is done by an activator protein called CAP, that is catabolite activator protein, which is a homodimer. It binds to the CAP binding site which is located adjacent to the promoter region of the operon, but the CAP cannot bind to the CAP site on its own. It needs the cyclic AMP. Cyclic AMP molecules bind with the CAP protein and form a complex. Then this complex will bind to the CAP site and induce or promote the binding of RNA polymerase to the promoter region. And so it will promote the transcription of these structural genes, which encodes for lactose metabolizing enzymes. Right. But the key feature is that this concentration of cyclic AMP molecules is closely related to the glucose concentration available in cell. How? Let's see here. If glucose concentration is high in the cell, then the cyclic AMP concentration will be low, and so it cannot bind to the cap protein, and so the inactive cap protein cannot bind to the cap site, and so the lactose metabolizing genes will not be transcribed. Now, if the glucose concentration is low, then cyclic AMP concentration will be high, and so it will bind to the cap protein, and the active cap protein now binds to the cap site, and ultimately the transcription of the structural genes will be carried out. Now here are few cases through which you can understand the exact mechanism of catabolic repression. In first case, when the glucose concentration and lactose concentration are low, then what will happen? Cyclic AMP concentration will be high because glucose is low. So the cyclic AMP will bind with the cap protein, and this complex will attach to the cap site, and it will induce the binding of RNA polymerase to promoter region. But lactose concentration is low, so lac repressor will bind to the operator region and not allow the RNA polymerase to carry out the transcription of the structural genes. So ultimately the transcription level will be low. In second case, when the glucose concentration and lactose concentration both are high, then what will happen? Cyclic AMP concentration will be low as the glucose is high. Then it will not bind with the cap protein, and so it will not bind to the cap site. But RNA polymerase can bind to the promoter region even though, because the lactose concentration is high. So the allolactose will bind to the repressor protein and it will not bind to the operator region, and so RNA polymerase will continue the transcription of these structural genes, but the transcription level will be basal level. What is basal level of transcription? When the repressor and activator both are absent, then RNA polymerase carry out the transcription of the genes on its own, and then the transcription is at basal level. Now, in third case, when the glucose concentration is low but lactose concentration is high, then what will be needed? Lactose metabolizing enzymes will be needed to be synthesized. So this cyclic AMP will bind to the cap protein and it will bind to the cap site, and it will induce the binding of RNA polymerase to the promoter region. Here, the allolactose will bind to the repressor again, and the inactive repressor cannot bind to the operator site. And so the RNA polymerase will continue the transcription of the structural genes, and the level of transcription will be high. Now in fourth case, when the glucose is high, lactose concentration is low, then what will happen? Due to high concentration of glucose, cyclic AMP concentration will be low, so it cannot bind to the cap site. And even if the RNA polymerase bind to the promoter region, the repressor has been there bound to the operator site, because lactose concentration is low, so repressor will be active and it will bind to the operator. So it will not allow the RNA polymerase to carry out the transcription process, and so the level of transcription will be low. So this is how the catabolite repression works. So this way the lactose operon particularly works under both positive and negative regulation. So it is the example of the operon system which works under dual control mechanism, positive as well as negative. Next time we will talk about the tryptophan operon in next video lecture. Okay, so see you in next video lecture. Please click like and share button if you like the video. Please subscribe my channel. Thank you once again.